Hello, my name is Alexei Lopez, and today I'm going to present about Taxol. And as you can see here, there's also another name synonymous with Taxol, which is paclitaxel. The paclitaxel, it's the same thing as Taxol, but paclitaxel is the generic name of the Taxol drug. Now let's get this presentation started. First, let's start with the mechanisms of action of Taxol or paclitaxel for cancer. And those will be a large majority reports indicate that paclitaxel arrested cells are in the metaphase and contain near bipolar spindles. Canonical images of paclitaxel treatment show cells with chromosomes aligned to the equator, although some cases or a few chromosomes have been reported to maintain or remain misaligned. Next is the paclitaxel-induced mitotic arrest occurs due to the activation of the mitotic checkpoint, also known as the spindle assembly checkpoint, the major cell cycle control mechanism acted during mitosis to prevent chromosome misaggregation. And here we're just repeating that paclitaxel is the genetic name for Taxol. Next, let's look at the side effects of Taxol. Side effects include vomiting, diarrhea, mouth sores, muscle pain, joint pain, numbness, tingling, burning, hands and feet, flushing, dizziness, and drowsiness may occur as well. Another side effect that may occur is the temporary hair loss. Another temporary side effect is the hair loss, but normal growth should come back after the treatment is over. And finally, but this is more rare, Taxol may also change the levels of blood pressure and the heart rate, but that's rare. Now let's look at the history of Taxol. Between 1960 and 1981, the National Cancer Institute, or the NCY, and the U.S. Department of Agriculture, which is the USDA, collaborated on the screening program and tested over 150,000 extracts from 15,000 species of plants to identify naturally occurring compounds with the anti-cancer activity. Samples from a single Pacific Jew tree, Taxus brevifolia, were obtained by the USDA botanist Arthur Barclay on the last day of his expedition in 1862. Mansuk won the inaugural wall, working under contract with the NCY at the Research Triangle Institute, received the tree Vibrifolia samples in 1964. Then, by 1967, they had isolated and identified the active ingredients from the bark of the tree Vibrifolia and named it Taxol, based on its species of origin and the presence of hydroxyl groups. Finally, by 1978, Taxol has shown efficacy in a subset of mouse tumor models, including the P388 leukemia, and it entered clinical trials by 1984. Now, some questions that I at least have about Taxol, and one of them would be, even if Taxol is being clinically tested to this day, can researchers and scientists find more efficient and safer drugs to cure cancer? And then finally, my other question would be, is a Taxol used to combat different forms of cancer, not just one type of cancer? How does Taxol differ, if it ever should, in combating each different specific form of cancer at the cell cycle molecular level? And to simplify this, my second question, what I'm actually meaning to ask is that for the drug Taxol, does the drug Taxol actually target the same things at a cell cycle level for all cancers, or let's say, for example, does Taxol target a different part of the cell cycle for, let's say, like breast cancer than it does for like another type of cancer like lung cancer? So I was just wondering that if the way that Taxol targets certain parts of the cell cycle is the same when it comes to treating all cancers, or it's different for every type of cancer. So that was my question, just to my second question just to clarify it. And that's the end of my presentation. And I hope you all enjoy my presentation. And thank you for listening my and or seeing my presentation.